Calculators. Lesson number 6. Growth. Growth in which irreversible change in mass, increase in size, volume and weight of any part of plant's body. It means quantitative increase in plant's body. Cell division, cell enlargement. Development, irreversible change in state. It means the qualitative change in plant body. Seedlings, vegetative maturation, flowering. Growth is a continuous process. Development is phase-to-phase process. Plant growth regulating compounds, natural and synthetic, natural found, natural and plant synthetic, human made. Both groups regulate or influence cell division, cell differentiation, root and shoot growth, senescence, plant aging. Plant growth regulators refer to natural or synthetic substances influence the growth and development. Classification of plant growth regulators on the basis of origin. Natural hormone produced by some tissues in the plant, also called endogenous hormones, IAA, indole acetic acid. Synthetic hormone produced artificially and similar to natural hormone in physiological activity, also called exogenous hormones. For example, 2,4-D NAA, naphthalene acetic acid. Classification of PGR on the basis of nature of function. Growth promoting hormones. Growth promoter increases the growth of plant, auxins, gibberellins, cytokinins, etc. Growth inhibiting hormones or growth retardant inhibit the growth of plant. For example, ABA, ethylene. Auxins derived from the Greek word auxin means to grow or increase. Auxins may be defined as growth promoting substances which promote growth along the vertical axis when applied in low concentration to the shoot of the plant. Auxins are synthesized in the stem and root apices and transported through the plant axis. Occurs universally in all plants. Active growth is equal to auxin production. Role of auxin. Auxin stimulates cell elongation, influence a host of other developmental responses, root initiation, vascular differentiation, tropic responses, and apical dominance. Development of auxiliary buds, flowers and fruits. Auxins in plant tissue culture are used to induce callus from explants and cause roots and shoot morphogenesis and parthenocarpy. Gibberellin have a regulatory function, are produced in the shoot apex, primarily in the leaf, primordial, leaf, bud and root system. Role of gibberellins: stimulates stem growth, dramatically stimulates cell division, cell elongation or both, and controls enzyme secretions. Involved in overcoming dormancy in seeds and buds. Used commercially in increasing fruit size of seedless grapes, stimulating and germination and seedling growth. Cytokinins promote cell division, found in all tissues with considerable cell division, eggs, embryos, seeds and germinating seeds, young developing fruits, roots supply cytokinins upward to the shoots. Interact with auxins to influence differentiation of tissues, may be used to stimulate blood formation. Cytokinin also modify apical dominance in stems and promote lateral bud growth. They also stimulate seed germination. This was the effect of cytokinins. Ethylene, growth retardant. Ethylene promotes ripening gaseous hormone produced in the actively growing meristems of the plants in, senescing, ripening or aging fruits, in senescing, aging or dying flowers, in germinating seeds, and in certain plant tissues as a response to bending, wounding or bruising. Ethylene as a gas diffuses readily throughout the plant. May promote leaf senescing and abscission. Leaf leaf fall increases female flowers in cucumbers. Economically will increase fruit production. Degreening of oranges, lemons, and grapefruit. Ethylene gas breaks breaks down chlorophyll and lets colors show through. Abscisic acid, growth retardant, induce stomata closing, inhibition of bud growth and shoot formation, widespread in plant body, moves readily through plant. Abscisic acid appears to be synthesized by the leaves, interacts with other hormones in plant, counteracting the growth, promoting the effects of auxins and gibberellins. Involved with leaf and fruit abscission, fall, onset of dormancy in seeds and onset of dormancy rest period in perennial flowers and shrubs. Abscisic acid is effective in inducing closure of stomata in leaves, indicating a role in the stress physiology in plants. X increases in abscisic acid following water, heat, and high salinity stress to the plant. Thank you. Techniques and culture conditions. Lesson number 7. Sterile techniques, clean equipment, surface sterilization of explants. Clean equipment. Successful tissue culture requires the maintenance of a sterile environment. All tissue culture work is done in a laminar flow hood. The laminar flow hood filters air with a dust filter and a high efficiency particulate air HEPA filters. It is important to keep the hood clean, which can be done by wiping it with 70% alcohol. Clean equipment. The instruments used should also be dipped in 70% ethanol and sterilized using flame or glass beads. Hands should be disinfected with ethanol before handling cultures in order to avoid contamination. Clean equipment. It is imperative to maintain exogenic conditions throughout the life of cultures, from egg plant to the production of whole plants. Entire experiments have been lost because of an episode of fungal or bacterial contamination at any stage of culture. Especially problematic are fungal contaminants that are propagated by spores that might blow into a wood from an environmental source. Therefore, it is important to work away from the unsterile edge of a laminar flow hood. Surface sterilization of explants. Plant tissues inherently have various bacteria and fungi on their surfaces. It is important that the plant be devoid of any. It is important to maintain hygienic conditions. Entire experiments have been lost because of an that's why. It is important that the plant be devoid of any surface contaminants prior to tissue culture, since contaminants can grow in the culture medium. Surface sterilization of explants. Plant tissues inherently have various bacteria and fungi on their surfaces. It is important that the explant be devoid of any surface contaminants prior to tissue culture, since contaminants can grow in the culture medium, rendering the culture non-sterile. In addition, they compete with the plant tissue for nutrition, thus depriving the plant tissue of nutrients. Bacteria and especially fungi can rapidly overtake plant tissues and kill them. Surface sterilization of explants. The surface sterilants chosen for an experiment typically depend on the type of explant and also plant species. Explants are commonly surface sterilized using sodium hypochlorite, household bleach, ethanol, and fungicides when using field grown tissues. The time of sterilization is dependent on the type of tissues. For example, leaf tissue will require a shorter sterilization time than will seeds with a tough seed coat. Wetting agents such as twine added to the sterilant can improve surface contact with the tissue. Surface sterilization of explants. Although surface contamination can be eliminated by sterilization, it is very difficult to remove contaminants that are present inside the explant that may show up at a later stage in culture. This internal contamination can be controlled to a certain extent by frequent transfer to fresh medium or by the use of a low concentration of antibiotics in the medium. Overexposing tissues to decontaminating chemicals can also kill tissues, so there is a balancing act between sterilizing explants and killing the explants themselves. Culture conditions and vessels. Cultures are grown in walk-in growth rooms or growth chambers. Humidity, light and temperature have to be controlled for proper growth of cultures. A 16-hour light photo period is optimal for tissue cultures, and a temperature of 22 to 25 degrees centigrade is used in most laboratories. I guess it is to 22 to 258 centigrade or 22 to 25 degrees centigrade. And next is a light intensity of 25 to 50 micromolar per meter per second per square meter per second. PAR, photosynthetically active radiation, is typical for tissue cultures and is supplied by cool white fluorescent lamps. Culture conditions and vessels. A relative humidity of 50 to 60% is maintained in the growth chambers. Some cultures are also incubated in the dark. Cultures can be grown in various kinds of vessels such as petri plates, test tubes, magenta boxes, bottles and flasks. Thank you.